everyone, my name is Katelyn Richards and currently I'm a senior biochemistry and molecular biology major here at the University of Massachusetts Amherst. I work in Professor Juan Lee's lab with a focus on plant epigenetics and plant biology. Epigenetics is the study of how organisms are altered by changes in gene expression such as by environmental stimuli but not changes to their genetic code. My senior thesis project is studying epigenetic regulations and vernalization response in the model organism Arabidopsis thaliana. Arabidopsis is a great model organism for studying this because it has a small genome. It's very well studied. There's many bioinformatic tools such as TARE available to us, and its genes are highly evolutionarily conserved, which means by understanding Arabidopsis, then its genes probably have a similar function in a related plant. The vernalization response is the process by which prolonged exposure to the winter cold triggers an epigenetic switch in a plant that causes many plants to flower in the spring. In Arabidopsis, this epigenetically switched gene is known as flowering locus C, or FLC. FLC is a floral repressor that is epigenetically repressed by exposure to the cold. Prior to vernalization, FLC levels are really high and the plant cannot flower. However, prolonged exposure to the cold triggers the vernalization response, which causes FLC levels to lower and the plant then flowers. The long-term goal of my project is to have a better understanding of the components that are sufficient to trigger the vernalization response in Arabidopsis and inhibit FLC. It is known that FLC is inhibited by the PRC2 complex, or polycomb repressive complex. I'm interested in studying three components of this complex that are upregulated in the presence of the cold: the protein vernalization insensitive 3, VIN3, and the two long noncoding RNAs known as COOLAIR and COLDAIR. It is hypothesized that by simultaneously upregulating COOLAIR, COLDAIR, and VIN3 in Arabidopsis thaliana, this would be sufficient for epigenetic silencing of FLC. So what's the importance of this research, you might ask? The societal and scientific impacts of my research are that by better understanding it in Arabidopsis, and because it's highly evolutionarily conserved, it probably has similar functions in other plants that use vernalization, such as many of our agricultural crops. By better understanding it in Arabidopsis and applying this research to agricultural crops, we could optimize their flowering time and thus have higher crop yields from the same amount of crops. This could help major problems like world hunger.